Hi, I'm Theodor Rostrup and I am CEO and co-founder of Atana. Today my presentation is divided into three sections. A quick introduction to Atana for those of you who just want to get the goodies rapidly, and then two sections about our two different business units for those who are more interested in details on what we are doing. So Atana was founded because we realized that we could probably contribute to increase the productivity in drug development by increasing the success rate of clinical trials. When we started, it was only 1 out of 10 drugs that entered clinical trials that reached the market. We analyzed what was measured and what was not measured, and concluded that nobody is really looking at the interaction under in vivo conditions. So either you basically look in vivo experiments in animals and see what effect a drug has and if it provides side effects or not, or you do mechanistic experiments in biochemical purified assays. Whereas we then try to bridge these two things. So we try to characterize the interactions under in vivo-like situations to really understand the mechanism of actions under the right conditions. And by doing so, our clients has been more than 4 times more successful in clinical trials than the industrial average. So, based on this success, we have two business areas right now. The traditional one, life science for drug development, and then we have also used our knowledge from that to establish a diagnostic area. In our life science, we have 2 legs. We are offering both analytical instruments, so our clients buy the instruments and the consumables and technical support from us and perform the work themselves. But we also have contract research, which means that the clients send the samples to us and we perform the experiments, do the analysis, and provide recommendations on how to proceed. Based on these experiences, where we, for instance, have done a lot of analysis in blood, we have during the pandemic launched our diagnostic platform, which is based on the same technology and based on the same concept to really understand interactions as they occur in the body. So how does the human immune defense system work, and thereby be able to see grade of immunity rather than just whether you have antibodies or not. So our diagnostic platform is generic. It basically works on any kind of disease you are looking for. But we have now focused on the COVID-19 assays. So, as I said, the reason why we founded Atana is that only 1 out of 10 drug molecules reach the market after entering clinical trials. What we're focusing on is to eliminate unwanted interactions, which is basically phase 1. So we can detect these unwanted off-target interactions and help our clients to eliminate or minimize them so that you decrease the risk of side effects. And we can also optimize the target interaction, so we select the right molecules and have them as efficient as possible in order to increase the efficacy in phase 2 studies. So that means that basically in the drug development process, we position us here right before clinical trials start. So we do optimization and validation of the drug molecules prior to entering the clinical trials. And how well have we succeeded? Well, roughly a year ago, we published a study on the 79 drug molecules that we have analyzed in our contract research program. Of these 79, we validated 30 after some optimizations and rejected 49. So most of them were rejected. Of the ones that we validated, 9 has so far entered clinical trials, phase 1, and 6 has proceeded to phase 2 studies, and 1 has concluded also the phase 2 study and is now entering phase 3 study. So this has been very successful. We also have one case where the molecule was rejected by us, but that one still entered clinical trials, and as we predicted, it wouldn't have enough efficacy, so it also failed in phase 2 studies. So if we sum all these, we can see that when we compare our clients' success in clinical trials, it's more than 4 times more successful than the industrial average. So how does this work? We use a technology called quartz crystal microbalance. And when we decided to found the company, and we decided that we should characterize the molecular interactions as they occur in the human body, one thing was that it was clear to us that one needed a label-free technology, because any modification you make on the drug molecule might also affect the interaction properties of it. So that was the first decision we made. The second was that we would focus on biopharmaceuticals, antibodies, proteins, Fabs, and so on. And the reason is that that's a more changing industry. It's a little bit newer, so it's easier to introduce a new technology in that area. The third thing was obviously that we should simulate in vivo conditions as much as possible. And one requisite for doing that is that you have real-time data. And why is that important? It's because if you want to characterize the interaction, you want to make sure that you only look on the interaction and no downstream effects. So any kind of secondary effects you measure might not really be influenced by the interaction. So if you want to optimize the interaction, you need to measure exactly that interaction and nothing else. So that was basically the prerequisites. So given that, we have 8 different assays. And the idea is that it should both be compatible with biochemical assays that is performed as standard today, and then it should add the more in vivo-like situations. So we can do everything from biochemical assays in purified buffers up to cells and tissues in sera and blood. And by doing these kind of experiments, we get basically 10 outputs. The most important is obviously affinity and the kinetic, the association and dissociation rate. But also very important is the number of active receptors on cells. And by comparing the results from biochemical, cell-based and tissue-based assays, and also comparing the interaction profile in salt buffers, in blood, in cell lysozymes, and so on, we get a comprehensive understanding of how the interaction is affected by different environmental parameters, and can hence help our clients modify their molecules so they are optimized for the given conditions they are supposed to act in. We can also detect a lot of fast and weak but frequently occurring off-target interactions. And by detecting them, we can also enlighten our clients on the need of removing them or minimize them, and thereby decrease the number of off-target interactions and hence the risk for side effects. So from that then, we have our two offerings within the drug development, the contract research, where we start with research intelligence. So basically together with you, we set up a project description to understand what information is needed and what is known before, what other technologies has been used, and then try to bridge the gap in information that is needed to get a comprehensive understanding. Then we use our technology and perform the experiments and provide reports and recommendations to our clients. Many of our clients also acquire their instruments, so typically then they buy the instruments and perform routine assays, and then also use our contract research for more advanced or when it's important with speed. And also a lot of contract manufacturing and quality control is done with our instruments because, for instance, in scaling up, it's not only important to look at affinity, it's actually very important to see that the kinetics is the same for the different production methods. So our clients are main interest of areas are the ones working within biopharmaceutical drug candidates and who want to validate and optimize the drug molecules prior to clinical trials, but also the ones who are in phase 1 and phase 2 and observe difficulties there, where we can actually go in and then identify, for instance, off-target interactions and see if that can be eliminated. And it's also an important knowledge for next research project to see how our technology can be implemented. From that, we have then used this experience of analyzing interactions in blood. And now, during the pandemic, there are a lot of different kind of tests, and most of the diagnostic technologies, they use technologies where you incubate the sample. They need enough time in order to detect something. They basically provide a yes or no answer if you have a level of antibodies above a certain level. And in order to detect, they use secondary antibodies or nanoparticles. And that basically just gives you the binary answer, yes or no, do you have antibodies? But it doesn't provide whether are you immune or not, or how immune are you. And that's where we try to contribute. So first of all, we do instant measurement. We don't have incubation. We follow the interaction in real time. As I explained previously, that's important. We get more information, not only if you have something present, but also the interaction profile of that, which can then be used to determine the level of immunity. We have direct injection, so no risk for artifacts there. And the overall combination of this gives an estimate of the quality of your immune antibodies, basically the neutralization capacity. So that is what I'm going to describe now. So what we do is that if you look on the left part, we have the virus. We take the spike protein and immobilize it on our sensor surfaces. So it can be the different variants, the Wuhan, Delta, Omicron, and so on. After that is immobilized, we take the blood samples, which can be both serum, plasma, or full blood, and flow over the instrument, where you will have a lot of different interactions. Everything that can interact with the spike will be detected here. After that, we stop providing the blood or the serum and flow over a buffer, and then we have the strongly bound antibodies or similar remaining, so we can get a readout of that. And then finally, we do the same as everyone else. We inject a secondary antibody to for instance, see how many IgG antibodies you have. And the reason we do that is basically in order to comply with the industrial standard, so you can compare our data with other technologies. So how does this look in real life? So what we do, looking at number one there, is that we have the spike immobilized on the surface. We take up a stable baseline. And this graph then, you can see the amount binding on the y-axis, and you can follow the time on the x-axis. And we have two different individs. So during 2, we inject the blood, let it flow over the surface and monitor the interaction. And in 3 then, we stop supplying the blood and look at the dissociation phase and how much is remaining. And then in 4, we inject the anti-IgG, anti-IgM, or so on to do the compatible with other things. So we then put this in a spider diagram. So number 1 is the total response, that is as the interaction occur. And as you will see it, and you can see here, it's different from different persons. Number 2 is then the amount of strongly bound, which can, for instance, be a combination of IgG and IgM antibodies. Number 3 is the quality, basically the dissociation rate. And number 4 is the amount of IgG antibodies. So number 4, that's equivalent to the standard technologies today. And I will show some data how we compare with other. And I will follow that with also look on the total specific response. So in our COVID-19 assay, we have, when we got our CE validation for it, we compared with the methods from Roche and Vaileo. So here we have 4 patients, 2 IgG negative and 2 IgG positive. And you have one that is very negative, you have one some is just below the cutoff, and you have one positive that is above the cutoff, and then you have one some is very positive with all 3 methods. So this is experiments performed in our CE marked assay. In the following graph, we take the same patients, but we look at it in our more kinetic assays. And as I said, we will look on 2 different aspects. So in these graphs, you see the same individuals before to the left and after to the right vaccination. And you can see in blue and light blue the IgG response for Wuhan and Delta. And so that is number 4 in the graph before. And then you can see in red and pink the specific full blood for Wuhan and Delta for the 4 individs. And what is interesting then is that if you look on before and after vaccination, you can, for instance, see that for individ number 1, the full blood response has increased relatively more than the IgG response for individ number 1, whereas for individ number 4, you can see that the IgG response has increased more than the full blood response. So you have different individual response for the 2 persons, as you can see here. And this is very interesting because we believe that this tells something both on how the quality of the immune system, but it can also provide information on at which phase in development is your immune response. So it's, for instance, likely that individual number 4 was exposed to COVID before vaccination, whereas individual number 1 wasn't. So individual number 4 has had more time to develop its immune response, and then of course we can all have individual responses. But if we then go and look on these immunity profiles, the spider web to the right here, we can see that we see to the left the IgG response. So the green one is individual number 4 and the blue one is individual number 1. And as you see in the graph, the IgG response is much higher for individual number 4 than number 1. You can also see to the right in this spider diagram the total specific immune response, which is then higher also for individual number 4 than number 1. What we also have done here is that we have analyzed the dissociation rate, so the quality, the neutralization effect, how well it can do that. And we can also see that the quality here is higher for individual number 4 than number 1. But on the top, you see the total immune response, that is during when the blood is present, when we measure directly. It's actually so that individual number 1 has a higher response. So what this means is that there are more things interacting with the surface for individual number 1 than number 4, but those items are not interacting very strongly. So this is an excellent tool both for vaccine development and to monitor how the immune system develops. And from that we can then, in the future, when a lot of studies has been performed, draw conclusions how well the immune system is developed for each individual. So the concept here is basically very generic. So we have our instrument, we have our immune assay kit, and basically you have the same chip, you have the same kind of reagents except from the antigen, which you can then immobilize easily different. So you can have either the different versions, Delta, Omicron and so on, or other diseases. But you have the same kind of assays, so everything is the same, so it's very easy to compare. And then of course we provide all the training and support for that. So our clients here are the ones who are working with immunology and virology research. It's vaccine development. For instance, we have Expression Biotech in Denmark, who will start their phase 3 development of their COVID vaccine. It's also good to look on therapeutic antibody development, because you can see how well, the same way as you look on the immune antibodies, the body's own, you can also test how well your therapeutic antibody interacts with different kind of antigens. And we also have several customers who use this for developing their standard IVD kits. So, thank you very much for listening.